So under this topic, what we're going to learn here is we're going to look at two, because when we talk about control and coordinations, we have two major pathway in terms of the coordinations between the organs. Okay, like what we know that we have the electrical signaling pathway and chemical signaling pathway. Okay, so same goes to the plants. They, even though they don't have the nervous system, they do have a certain mechanism to allow the transmissions of the action potential. Means that they still will be able to communicate by using the electrical signaling pathway. It's just that they don't have a specialized cells like neurons that we have to transmit or to generate the action potential. So they do have their own way to do this. Okay, so the first part for today's lessons, we're going to look at the electrical signaling pathway, where involve okay, the rapid response of venous flytraps to stimulate the hair, to respond to the stimulations of the hair, okay, on the lobes of modified leaf, and explain how the closure of the trap is achieved. Okay, so this part will be electrical signaling pathway. And after that, we're going to look at a chemical signaling pathway where it involves two kinds of hormones or plant growth regulator, plant growth regulator. So one is the auxin. Another one is gibberellin. Gibberellin, we have talked about it a little bit when we deal with the transcription factor, right? So today we're going to look at it in detail about the gibberellin. And we can look at the effect of the auxin in the elongation growth by stimulating the proton pump to acidify cell wall. So we're going to look at how the auxin transportations will allow this elongation growth. And to be careful in this case, or to be more precise here, please write down the elongation growth here is actually refer to the cell elongation. Be careful, I mean, when you write, okay, because auxin only involved in the cell elongations, but later we'll look at gibberellin. Gibberellin involved in both cell elongation as well as the stem elongations. Okay, so auxin only involved in the cell elongations. Okay, and then we're going to look at the to describe the role of gibberellin in what? In the germinations of the wheat or barley. Okay, and we're going to look at how gibberellin break the dormancy, okay, by activations of the gene. Okay, so we're going to look at this. And last part, we're going to look at the role of gibberellin in the stem elongation. So when you talk about gibberellin, so involved in the stem elongation rather than compared to auxin. Auxin only cell elongation, but gibberellin involved in the stem elongations. And we're going to look at the dominant allele, big LE allele, and also the small LE allele. So what is the genetic behind how gibberellin will be able to cause the stem elongations? Okay, so without delay, let us look at the electrical communication in plant first. So we do have the plant cells, and we do know that plant cells have this electrochemical gradient as well. Okay, they can have electrochemical gradient. So basically due to the distributions of the charge. Okay, so like what we do know that if you pump the proton out of the cells, then outside the cell will be positively charged, inside the cell will be negatively charged. Or they can use the sodium potassium pump, constantly pump 3 sodium ion out and pump in 2 potassium ion. Again, we'll be able to generate this, what we call the electrochemical gradient. Okay, so this electrochemical gradient across the cell surface membrane in the same way as what we animal cells can do, and they have this resting potential as well. So as in animals, so the plant action potential are triggered when the membrane is depolarized. Okay, so it depends on how the depolarization takes place. As I said that previously, when we want to create action potential, there must be a rapid change in terms of the distributions of the charge. Like for example, we do know that in neurons, our neurons, internally we have negatively charged, externally we have positively charged, and because of action potential generated, there is a rapid influx of the sodium ions into the cells, correct? Okay, so in some species, there's some responses to the stimuli by coordinating the action potential. For example, our sensitive plant called Mimosa pudica respond to the touch, right? One type of plant, when you touch it, it will close the leaf, right? So this kind of the plants, actually, they communicate by electrical communication because the response is very, very, very fast. Okay, so if we use the microelectrode inserted into the leaf cells, we can detect the change in this potential difference or membrane potential, they are very similar to action potential. So some of these plants, okay, the depolarization result from the outflow of negatively charged chloride ion. So a little bit difference, okay, because if you look at in terms of our mammal or humans, okay, our depolarization characterized by what? The influx of sodium ion. So making it become less negative, right? The membrane potential become less negative. But in some plants, okay, there are depolarizations in plant. Their depolarization actually is the exflux, or the chloride ions actually is the one that move out. So again, same what we call this outcome. So less negative again. Okay, so when chloride ion move out, it become less negative. So again, it will from less negative become more positive and then change back to negative. So again, this is how we generate the action potential. Okay, so the repolarization is achieved by outflow of the potassium ions, similar to the humans or mammal. Okay, so plants do not have a specific nerve cell, but they can actually transmit a wave of electrical activity very similar to the neurons of the animal because the presence of the plasmodesmata. So how this plasmodesmata actually functions here? Now let us look at this. This is the plant cell. So where the plant cell wall. Okay, so we do know that plants, they have the plasmodesmata. Yeah, let me draw the plasmodesmata. Okay, then they do have the membranes. Okay, so this is the membrane. So you can see that plasmodesmata allow the continuity of the cell surface membrane. Can you see that? And this continuity allowed the action potential to be transmitted. And again, we do know that, okay, that for example, if we trigger any action potential here, this action potential can actually transmit it. Can you see that from one cell to another cell? For example, this part, continue. Okay. Now, if that's even though we say that, okay, they can transmit the action potential, the rate or the how fast this action potential can be transmitted definitely slower compared to the animal. Why? Because they don't have a specialized cells. They use this plasmodesmata to transmit the action potential across, okay, two different cells. Are you clear? Okay, so the action potential generate lasts much longer and then travel more slowly compared to the animal neurons, because it's not specific one. Okay, so we're going to study this by using the Venus flytrap. Okay, so before we start, let us look at the structure of these carnivorous plants, where we call it the insect eating plant in this case. And they're not really eat the plants, but what they do, they actually have the enzyme to hydrolyze these insects, okay, when they trap the insects. So you have to look at this. They are the leaf, actually. They are specialized leaf. Can you see that? The leaf, actually, each lobe of them, this is a mid-ribbed. Okay, this is a leaf. This is a mid-ribbed. So each lobe of the leaf, they have these 3 sensory hair. A stiff sensory hair here. Okay, and then you can see that outside the edge of the leaf, they do have this stiff hair as well. So both hair actually have different functions. So this one actually is a sensitive hair. This stiff hair actually something like the lock, so they can interlock each other and trap. We try to imagine when they close together, and then this the stiff hair will interlock together and traps the insect inside. Okay, so now we can see that the specialized leaf divide into 2 lobes, and each lobe will have this red and has a nectar secreting glands here around the edge. Okay, this the edge of the leaf here, and to attract the insect. Okay, so the nectar. So each lobe will have 3 stiff sensory hair that respond to be the touch or deflections here. So the outer edge of the lobe also have the stiff hair that can interlock to trap the insect inside. And the surface of the lobe also contain the glands that secrete the enzyme that can digest the trapped insect. So what actually happened here, it depends on the touch. So the touch, let's say for example, the touch on one hair, it will trigger this action potential. But again, trigger of one hair won't cause the closure of the leaf. But trigger the second hair within 20 to 35 seconds. Okay, let's say I touch you one, I touch another hair, another one. Within 35 seconds, 20 to 35 seconds, what will happen here? Immediately the trap close. Are you clear? So it means that the touching of this hair actually generates the actual potential. Okay, the touch of a flies or other insects on the sensory hair on the inside of the folded leaf or the Venus flytrap stimulate the action potential that travel very fast across the leaf and cause the leaf to cross to fold over and close and trap the insect. Okay, so I'm going to show you guys the video clips about this, how the trap close and trap the insect. Okay, so this process actually is very how to say that? Okay, there are many theory talking about this. Okay, so we are not going to learn in the detail how the action potential transmitted. So basically, if you draw the leaf, this is how the leaf look like. This midrib. Okay, sorry. Okay, we use sideways this midrib. Okay, so this is the lobes. Okay, two lobes with the stiff hair, and within the leaf, they do have these three stiff hair on each leaf. Correct or not? So first thing here is this actually we have two theory talking about this. Two theories. Okay, one is called the tension theory. The tension theory basically means that it's something like, I think last time, do you guys play this before or not? Okay, like a ruler or the watch. Okay, they're actually something like this, the watch. Okay, this is your watch, and you hit the watch onto your hands or your wrist. What will happen? It was turn and curve. It's like something like spring. Okay, and when you hit on something, then it start to curve. So this leaf actually already have their own tension already. Okay, so it means that we just need to trigger this tension, then it's going to close. So how to trigger this tension? So basically, first thing we're going to touch. Okay, it's sensitive to the touch. So the touch on the sensory hair, okay, 2 times. So how these 2 times? These 2 times take place within 20 to 35 seconds. Right? 20 to 35 seconds, what will happen here is either you touch on the same one or touch on the different ones, then it immediately will close. Are you clear? So you can see that it starts from convex to become concave. In this case, you can see that's now a trap. Okay, this midrib. Go and trap the insect inside. Are you clear? But at this moment, this the stiff hair outside, the stiff hair here is not closed totally yet. So that's one of the mechanism. Why? Because if the small one, it will be able to escape, because when it close, I mean, try to imagine, okay, when it close, it will take about 7 to 14 days to complete the hydrolysis process. So if let's say the small one, you try to imagine you spend so much of time, but your food is only a small portion you want a big portions here, right? So therefore, the closure of this trap is not fully sealed yet, okay? But the thing here is you try to imagine the insect inside, when it closes, they try to escape, right? So the more they touch on this sensory hair, the more tightly this stiff hair actually going to interlock, trapping this insect. So when insect trapped inside already, what will happen? Insect even try to find a way to move out, right? So what will happen here, the third thing, when they touch more the hair, then it will stimulate the release of enzyme by exocytosis. So the leaf surface will secrete the enzymes. This enzyme going to hydrolyze the insect. Okay, now the second theory that I'm talking about here is due to the auxin. Now, auxin actually will cause the cells elongations, okay, the swelling of the cell become bigger. So this auxin also will cause the entry of the water molecule and then cause the cells to expand and then curve up to trap the prey or traps this insect inside. Are you clear? So basically three. First, okay, three processes here. First, the touch. So the leaf sensitive to the touch. How many times? 2 times within 20 to 35 seconds. It can be one sensory touch, 2 times sensory hair touch, two times, or 2 hair. Okay, but within 20 to 35 seconds, then it close. But the closure first is not tightly closed yet. Okay, the stiff hair actually close it. So allow those small insect to escape and then open up again. So okay, to wait for the big insect. Okay, but if a big insect continue to move inside, it will cause the Venus flytrap to close it tightly. And then because the movements of the insects, then it cause the release of the enzyme by exocytosis because of the calcium ions. Okay, it trigger the entry of the calcium ions. Okay, against calcium ion always involved in the exocytosis. Okay. So in this figure, we can see that figure 19.46 show the leaf of Venus flytraps, have a group of stiff sensory hair in their center. When these are closed or touched, the leaf respond by closing, trapping whatever was crawling over them. Okay, so you can try the questions now. Okay, so the touch of the fly, okay, of a fly or other insect on the sensory hair, okay, the sensory hair, okay, on the insides of the folded leaf of the Venus flytrap stimulates the action potential that travel very fast across the leaf. Okay, why? Because of this plasmodesmata allowed them to cross. If no plasmodesmata, then they won't be able to transmit this action potential because of the cell wall there. Okay, so the deflection of sensory hair activate the calcium ion channel in the cells at the base of the hair. So this channel opens so the calcium ion flow in to generate the receptor potential. It's that because now it's slightly different. It's slightly different from the how we have the sodium ion enter, but this one actually activate the calcium ion channel. So this one will generate action, right? This receptor potential first until it reach a threshold. So how do they reach threshold? They reach a threshold when the hair, okay, if 2 of these hairs stimulated within a period of 20 to 35 seconds, or 1 hair is touched twice within the same time interval, again, the action potential travel across the tract. Okay, they leave that. So when the second trigger takes too long or occur after the first, the trap will not close but reset. Okay, a new time interval start again. So I mean, if we touch it once and then we wait until after 40 seconds, touch a second, no issues because it's going to reset. In more than 35 seconds, you're going to reset it already. Why reset? Because the receptor potential, try to imagine, have to reach a threshold, right? So, but if you don't touch a second time, the receptor potential cannot reach the threshold. So therefore, it reset, it go back to the resting potential. So within these 20 to 35 seconds, understand the concept like what we have, right? The receptor potential reach the action potential, as the receptor potential reach the threshold potential, then we will have the action potential. Okay. So if a hair is deflected a third time, then the trap will still close. Why? If it deflected a third time, so depend on the second time and third time. As I said, that's now the first time, let's say 0 second, and second time by rights, this duration must be within 20 to 35 seconds. But now, if I touch it at 40 seconds, so means that the trap now reset. Correct or not? So we don't talk about this one already. So from now to the third time, this second time touch to the third. If the third one touch, let's say for example at 50 seconds, so the durations, okay, let's say okay, 65 seconds for example. So they touch the second time. So within this is less than 20 to 35 seconds, right? So means that this one will cause the closure of the traps. Okay, so if hair is deflected a third times, then the trap will still close. The time between the stimulus and response is about 0.5 seconds. So look at this, 0.5 seconds compared to us. To compare to the animal, we have the response times roughly about light. That one action potential to be generated is about light 1 to 5 milliseconds. So this is 500 milliseconds. So means that it's still very, very slow if we compare to the animal response, okay, to this action potential. So it takes a trap less than 0.3 second. Again, it's quite long, 0.3 second. Basically mean 300 milliseconds. Yeah? Look at it, okay, to close and trap the insects. So the lobes of the leaf bulge upwards when the trap is open, so it's convex. So this is how the first original. Okay, so convex, cannot. Okay, you can look at convex, the leaf, right? When it opens, so they are convex in shapes. No one is quite sure how the trap closed, but it's noticed that the lobe rapidly changed into the concave shape. So it means that from the convex into the concave, so that it can trap. Okay, can you see that? We are referring to the inner surface, become concave. Okay, so bending downwards so that the trap snaps shut. This happened too fast to be simply a result of the water molecule movement from the cells on the top of the lobes to the cell underneath. So basically means that from the top here to move downwards so that it can curve. Are you clear? So instead, it's likely to be a rapid change occur as a result of release of elastic tensions in the cell wall. So something like the ruler or the watch that you snap it on your wrist, then it start to wrap onto your wrist, right? So this is one of the possible mechanism. But in our syllabus, we need to know about the result of the movement of the molecules here because of what? Because of the auxins, okay? So when it comes to auxin, we're going to learn about this again, how these water molecule enter into the cells due to the actions of the auxin. So here I won't discuss it, okay? However, the trap is not completely closed at this moment. So to seal the trap, it requires ongoing activations of the trigger of the hair, okay, by the trap prey. Unless the prey is able to escape, means a small one, if it further stimulates the inner surface of the lobe, thereby trigger the further action potential. This forces the edge of the lobe close together, sealing the trap now to form an external stomach in this case, which prey digestions occur. So further deflection on these sensory hair by the trap insect stimulate the entry of the calcium ion. Again, highlight these calcium ions. So these calcium ions now stimulate the exocytosis. Still remember, calcium ions most of the time involved in the exocytosis, like in the synapse. Cannot exocytosis. So again, calcium ion. So these calcium ions stimulate the exocytosis of the vesicle containing the digestive enzyme in a similar way to their role in the synapse. Can you see that? Synapse exocytosis of the neurotransmitter, but here release digestive enzyme. So the trap stays shut for about a week for digestion to take place. When the digestion is done, the cells on the upper surface of the midrib grow slowly so that the leaf reopen and tension build in the cell wall of the midrib, so the trap is set again. So how the tension can be generated or is set, we do not know at this moment. But we look at it, it's appear that there is a tension that actually set on the trap. Okay. So what are the adaptations here? Because this is a common question asked. Highlight. So Venus flytraps have two adaptations to avoid closing unnecessary and wasting the energy, because the regenerates the action potential and everything require the ATP. Okay, so first, the stimulation of a single hair does not trigger the closure. This is probably to prevent the trap closing when it's rain or when a piece of debris fall onto the trap. So try to imagine it raining. Okay, so when the drain dropped, okay, onto it, okay, it triggered the hair might not be within 20 to 35 seconds. So it continuously to reset the timer. Are you clear? Okay, unless it's heavy rains, it means a different story. But again, it tried to prevent the unnecessary closure. Okay. Second, the gap between the stiff hair, okay, that form the bar of the trap allow very small insect to crawl out. Okay, so therefore the plant won't waste the energy digesting a very small meal. So these are two very effective way, okay, to make sure that the leaf close won't because of the unnecessary, like for example the rain or the debris, and also the small insects. Okay, so with this I have done for the first part of today's lessons, which is electrical signaling pathway.